This is our third and last video for the neurons and synapses section. This is section 6.5. And I'm going to go away again. Toodles. See you next time. Bye. So this part of the video, this part of the lesson rather, this video will focus on connecting points in the nervous system where one nerve cell may communicate with another cell. And they do that at these little connecting points. You see where this cell talks to this cell, little connecting points where they come quite close to each other, but don't quite touch. And that part where the cells are very close, but don't quite touch, that's called a synapse. So the rest of this is going to focus on what's called a synapse. So we have one cell here getting very, very close to the next cell. So this is the dendrite of the next nerve cell. And then this is the axon terminal of the last cell. And it comes very, very, very close, but doesn't completely touch the next cell. It forms what we call a synapse. These are junctions between cells in the nervous system. So, first up, those synapses can occur between two neurons. So you could have one nerve cell talking to another nerve cell. Or you could have one nerve cell or neuron talking to a receptor cell. So these are the cells that are involved in actually detecting changes. These are involved in sensory reception. Think about your eyeballs, for example. Or we could have neurons linking up to an effector cell. So this would be something that causes a response. So you have a nerve cell connecting to a muscle that makes you move. There's a response there. You could have a nerve cell linking up to a gland, and that would cause the gland to release some hormones or something. That would cause a response. So again, nerves can connect to nerves, they can connect to receptor cells, or they can connect to effector cells. Well, no matter what, we're going to have and what happens is the presynaptic neuron is going, so that's the one, before the synapse. We have the presynaptic neuron, and then we have the postsynaptic cell. No matter what the case is, the presynaptic neuron is going to be stimulated, right? So there's going to be an action potential coming down this axon, making it all the way to the end. And at that end, the presynaptic nerve cell is going to get depolarized, right? The action potential raises up, becomes depolarized, and then this cell is going to communicate. And the way that these nerve cells communicate is with chemicals called neurotransmitters. IB wants you to know one type of a neurotransmitter, which is called acetylcholine. And so we're going to use this example of a synapse, which is called a cholinergic synapse, which uses the neurotransmitter called acetylcholine. So keep in mind that as I'm describing this, this is just one type of synapse, but there are other types. And so here's how a cholinergic synapse works. First of all, an action potential has come from the nerve cell. It got stimulated, it was told to go fire. The action potential now arrives, boom, and we're here. So the action potential has made it all the way to the end of the nerve cell. This nerve cell has been depolarized. And what that does is it is going to signal a new voltage-gated channel. Remember we had voltage-gated ion channels before? Here's a third voltage-gated ion channel, but this one is calcium. So they have voltage-gated calcium channels right at the end of this cell, just in the synapse. And that calcium is going to basically signal these little vesicles. They're called synaptic vesicles. And inside that synaptic vesicle, that's where all of our neurotransmitters are located. So the calcium then signals neurotransmitter vesicles to move, to migrate toward the edge, toward the membrane, right here. And so this vesicle fuses with the membrane, releasing the neurotransmitters into this empty space called the synaptic cleft. Then the neurotransmitters diffuse across the synaptic cleft where they bind with receptors on the postsynaptic cell, and that is going to stimulate an action potential in the postsynaptic cell. So here it is again in writing. Calcium influx signals synaptic vesicles to then fuse with the presynaptic membrane, releasing neurotransmitters. The example that IB wants you to know, these are going to be acetylcholine neurotransmitters. So this green blob, that is acetylcholine, which then is released by exocytosis. The acetylcholine, abbreviated as ACH, diffuses across the synaptic cleft and binds to these protein receptors on the postsynaptic membrane. The binding of the ACH to these receptors causes an opening of sodium channels, which will stimulate this postsynaptic cell, bring it up to its threshold potential, and then the voltage gated sodium channels open, and this cell can do its action potential, just like we talked about in the last video. Then, after that acetylcholine has is binding to this receptor, it needs to be broken down because we don't want this postsynaptic cell to be stimulated over and over and over and over. We need to break down that neurotransmitter so that ACH detaches from the receptor and an enzyme is going to break it down. And because all of this stuff happens with a neurotransmitter called acetylcholine, we call the whole synapse a cholinergic synapse. There are other neurotransmitters in your body, and so those synapses would get other names. IB wants you to know the example of a cholinergic synapse, which happens to use acetylcholine. Now, let me back up. Now, a cholinergic synapse with acetylcholine, these are usually involved in movement. So this postsynaptic cell might be a muscle cell or something. So these cholinergic synapses are involved in you coordinating your movements. Okay, so that's basically what IB wants you to know for the cholinergic synapse. So if you can go through numbers 1, 2, 3, 4, 5, 6, and give me some annotations here, then you're in solid shape. All right. Then we get to the application. There's a lot of vocab here. Sorry. So IB also wants you to know this example of a pesticide called a neonicotinoid. These are pesticides that basically I'm gonna switch back and forth on the slide. So if you want to pause and get your notes down and then listen, that might be good. So what these pesticides do is they block the transmission of acetylcholine by binding to those receptors. So what those neonicotinoids do is they sneak in here to the cleft. These are little chemicals that we spray on insects. And then these neonicotinoids sneak into this cleft and they bind to these receptors. And if they're binding to that receptor, well, the neurotransmitter can't get in there. Yeah. So normally acetylcholine gets broken down by this enzyme called acetylcholine esterase. But those neonicotinoids are not broken down by the enzyme. Therefore, the effect of those neonicotinoids is irreversible because they bind here, they detach, and then the enzymes can't break them down, so then they can attach again and basically shut down this cell. And if this cell is a muscle, they have now shut down a muscle cell, and that poor little insect is going to be paralyzed. And that paralysis is irreversible. Therefore, usually they can't move, and that will, of course, lead to death. Now, why do neonicotinoids work on insects but not humans? Well, technically, they could actually affect mammals, but insects have so many more cholinergic synapses that the effect on humans is negligible, whereas on an insect, it's enough in small amounts to permanently paralyze them. Okay. Whoa, 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 whoa. Let me pause here. That is, excuse me, that is the big content from this section. So, can you describe the function of a cholinergic synapse? Can you describe the way in which a neonicotinoid pesticide will interrupt the normal functioning of this cholinergic synapse? And then I'm going to wrap again with another exam question. And this question reads basically this: what are the biochemical nature and function of the myelin sheath? Take a second, pause the video, think it over. All right, let's take them one by one. The myelin sheath is mainly protein, which allows growth of the axon. No, the myelin sheath is not mainly protein. B, the myelin sheath is mainly protein. No, it is not mainly protein. It is neither A nor B. Okay, so the myelin sheath is mainly lipid. Yeah, okay, cool. So it's either C or it's D. So does the myelin sheath function as an energy source, or does it function as a saltatory conduction? So no, C is the right answer. The myelin sheath is mainly lipid, which allows for saltatory conduction. Remember that saltatory conduction is jumping from node to node to node rather than a continuous flow. One more. This is another picture of a synapse. We've got our presynaptic cell. our postsynaptic cell. I'm going to encourage you to pause the video, read the question, give it a shot. Go ahead and give it a shot. Okay, I'm back in. I'm going to discuss the answer now. So, how do neonicotinoid pesticides cause paralysis and death of honeybees? Okay, honeybees are insects, and we know that neonicotinoid pesticides are designed to work on insects. So, what do? They destroy one. Okay, one is a vesicle with the neurotransmitter acetylcholine inside. No, they don't destroy that. No, it's not that. B, they bind to number two. Okay, so in this cell, it looks like these little blocks have these little dots binding to them. So this must be the acetylcholine receptor. So the acetylcholine is binding here. And yeah, B is the right answer. The acetylcholine normally binds to the receptor, but instead the neonicotinoid binds there and shuts down this cell. So that B is the right answer. Let's talk about C and D though. C, they inhibit the release of number 3. Well, they don't inhibit the release of the neurotransmitter. Instead, they block the function of the neurotransmitter. So the cells may still be releasing acetylcholine, but it won't do anything because the receptors are jammed up. D, they block the activity of number 4. What is number 4? Right, it's the mitochondrion. So this is a mitochondrion. No, they have nothing to do with mitochondria. So that's the end of that. So if you can go back and explain synaptic communication, you're in good shape. All right, I'll end the video here. And jump in right here. Oh, wrong one. I'm gonna end this video. I hate this so much. So that's it. Go back and read the chapter, and best of luck.